So I'll just briefly introduce Dongyuan and his work. So Dongyuan is currently a third-year PhD student in the bioinformatics program and in my lab. So today he will introduce our new work, which is still unpublished, but we're trying to wrap it up, which is about a new statistical framework. And I would say its biggest advantage is that it's an all-in-one framework. It can handle multiple cell heterogeneity structures, including continuous cell trajectories, discrete cell types, and spatial information. So under this framework, we could fit that statistical model to real data. So using the fitted model, we could do inference of parameters we are interested in, such as differential expressed genes. And also using the fitted model, we could simulate a synthetic data, which will look like real data. So I think this is a fantastic work. And so hopefully Dongyuan can introduce it to our broad audience here so we can have more users. Thank you.
Yeah, thanks for the introduction from Jessica. Let me share the screen. Can everyone see my screen right now?
Yep.
Thanks. So hello, everyone. My name is Dongyuan, and today I'm going to talk about our new work, IC Design 3. So we call this as an all-in-one statistical framework, which can generate realistic single-cell data and infer different cell heterogeneity structure. So here I will give a high-level introduction about this problem. So the challenge for nowadays single-cell data is that they are becoming more and more complicated. So we try to summarize the complexity of nowadays single-cell data into 3 dimensions. The first one is the heterogeneity structure. So this is usually the most interesting thing people want to know. Usually we can have cell types, so this can be known or latent, so you can infer them by clustering. You can also have continuous trajectories. This is usually latent variable because you don't know the actual time of cell is, so we can use the pseudo time inference to get an approximation of this latent variable. And we can also have spatial location. These are true observations because they are measured by experiments. The second dimension is the design of experiments. So we can have multiple batches. So here we have the so-called batch effects. Between different experiments, there are some technical variations, and people usually don't want to have those kind of variations. On the other hand, we can have multiple conditions. So in such kind of cases, the differences between different conditions are usually biological meaningful. So people are interested in those kind of differences. In the third dimension, we also have the feature measure. So we can have gene expression. This is the most familiar type we usually use. For example, single-cell RNA-seq and the spatial genomics, they are trying to measure the transcriptomics. And we have also the chromatin accessibility. So we can have SC-ataxic or snare-seq. They try to measure the openness of the chromatin of the genome. And we can also have many other different technologies which measure different features. For example, we have CytSeq. It can measure the surface protein, and also every day we have new technologies for single-cell genomics. So before we talk about the SC Design 3, we need to talk about SC Design 2. Obviously, this is a prior work for SC Design 3. So this is just published last year on Genome Biology. And the SC Design 2 can simulate cell types while keep the gene correlation of each cell type. So the limitation of this work is that it only works for single-cell RNA-seq and works for discrete cell types. And also for this model, although it is kind of a probabilistic model, we cannot get the likelihood. The likelihood is very useful because if you want to perform model selection or statistical tests, then you must have the likelihood available. So today I'm going to talk about the SEDANS3, and here I will give a brief introduction to it. So SEDANS3 model both the marginal features and the feature-feature correlation, and it is a complete probabilistic model with likelihood available. And the most exciting thing for SEDANS3 is that it unifies all kinds of single-cell data under one model. So what can we do for this model? What can we do by using this model? We can have a trajectory and spatial data. So as far as we know, this is the first simulator which can simulate such kind of data types based on our reference data. And we can also simulate the batch effects and the conditional effects. And we can also use multi-omics, for example, taxic, static, and other things. Please note that these three dimensions, they can have interactions. So you are possible to have like trajectories between different conditions, or like different batches from different ataxics. So this is completely available. And we call this SEDANS3 as an all-in-one solution to single-cell data. The first and the most important usage for this is to do the data simulation, because it can simulate any kinds of single-cell data based on users' specification. And the second usage is very interesting. It can help you to do the model assessment or model selection. So if your covariate is not observed but inferred from your data, for example, pseudotime or cell type by clustering, you probably want to know that the quality of your inferred pseudotime. For example, does it describe my data well enough? So here we can use the SEDANS3 to evaluate the quality of your latent structure. For example, you have pseudotimes from different pseudotime inference softwares, and you can use SEDANS3 to compare them and to choose the best one. And you can also use to do a model, choose a model explaining your data better. For example, you may wonder like if the gene-gene correlation is on the population level, or I should use a cell type specific gene-gene correlation to describe my cell population. So you can also use SEDANS3 to answer this question. And the third one is a statistical test. So SEDANS3 can also perform the conventional differential expression test. And it can also help you to do the distribution choice. Although this is usually the interest for statistician. For example, you can choose like if you want to use Poisson or negative binomial to fit your counts. So here we come to the methods part, and I will skip most of the details of this model, but I will introduce a very brief idea of it. So the input data of the SEDANS3 model are three things. The first thing is the cell by feature matrix. If this is transcriptomic data, then this is just like an expression matrix. But for other kind of technologies, for example, ataxic, you need to convert your genomic data into like a peak by cell matrix. But as long as your input data is a matrix, then this is okay. And the third thing is the cell-wise covariates. So this is per cell information provided by users. For example, cell types, spatial locations, batches, conditions, and etc. So this will be treated as a truth for this model. For example, if you provide 3 cell types, then this model will think these 3 cell types are the truth and perform model fitting. And the third thing is other model setting. For example, what the correlation structure is, and you can also input a different cell number. So by default, the output will be the same number of your input data, but if you want to have a different cell number, this is totally okay. For the model fitting part, we have 2 parts. The first one is a marginal model fitting. So we will use each regression models for each gene. And the second thing is the correlation part. So after we get the distributions for each individual genes, we will use a Copula model to modeling the correlation structures. So the basic idea for the Copula model is that we convert the marginal distribution into its corresponding quantiles. Then for each marginal, you now have a quantiles from 0 to 1, and you just try to modeling the correlation based on this quantile matrix. The outcome of the SEZ3 are also the three things. The first one is a new feature matrix, and the second one is a new cell-wise covariate matrix. If the cell number is different to the original cell number. And the third one is a model AIC, which is very useful for perform model selection and model assessment. So here we want to say something about how to use SEZ3 for model assessment and model selection. So we approach two different ways for this purpose. The first one is a very conventional way. So since we have the AIC, or it's just kind of penalized likelihood, we can just use it to do this. And the good thing for this is that we just need to fit the original data once. But the bad thing is that sometimes AIC cannot reflect the best model you want. So here we approach the second way. The second way is quite a novel way, and it is the model selection based on new data simulation. So here we have the assumption, if your model is better, for example, if your pseudotime is more accurate, then the simulated data should be more similar to the real data. So in other words, a better generative model can generate more realistic data. For this purpose, we need to have this train test split, because we don't want our model, it's just like an overfitting model. And we use the local inverse Simpson index to measure the similarity between the simulated data and the original data and the test data. And we can also use the 2D visualization on low dimensional space, for example, t-SNE and UMAP, to check if the simulated data is similar to the original data. I have to say the two ways are roughly consistent with each other, but with some like minor discrepancies. So here we come to the results of our SEZ3 framework. The first result I want to show here is an example for bifurcating marrow development data. So this is a continuous trajectory data with two lineages. So you can see the first half is one lineage and the second half is the other lineage. And here we only show the first lineage by showing the continuous pseudotime of this lineage. So from dark blue to yellow, the pseudotime is increasing. And here we can see, compared to the real data, our simulation framework works quite well by generating a very realistic simulated data. And we calculate the LRSI value, which is almost 1.9. So this is a very good value, which means that, so basically this LRSI value means if you overlap these two figures together, what is the chance you observe, what's the chance of each individual point you observe a point from another, from two batches or just from one batches. So if the data are perfectly similar to each other, then this value should be close to 2. And based on our observation, even you use a training set and test set to calculate this value, it is usually just 1.9. So this means that our simulated data is very similar to the real data. So the second example is for the spatial data. We try to simulate the spatial data generated by 10x Visium, and this is a brain example. So here we only choose one gene to show this because for spatial data, usually we care more about the plot on the spatial map. And here we can see our simulator also did a pretty well job for simulating the real data. We can see the basic spatial pattern has been preserved for our new simulated data. A not very good thing is that here the boundary is not as clear as the real data, but this is what we can expect because here for the spatial data, we will use a 2D smooth surface to approach the real data distribution. But in general, the results looks pretty good. In the next examples, we try to show the results of 2 different batches of the same biological samples. So this is PBMC cells, which measured by two different 10x technologies. So I think they are 10x versions 2 and version 3. And here we can see our simulator also generates the very realistic results compared to the real data. And we can see all the cell types are preserved in the new data sets. And we also show the batch effects between the 2 technologies. In the last case for the simulation part, we show the assay ataxic data, and it also measures the PBMC. You can see here we have many different cell types. And here the LISI value is also very high, which means that we simulate the data quite well. So if you observe the data more carefully, you can see here our cell types, the boundaries of our cell types are kind of more clear than the real data sets. This is because our simulation framework is decided by the input covariates. So you treat those covariates, those cell types as a truth. So the simulated data will try to generate those simulation based on your covariates. So therefore, it will kind of make the differences between different cell types larger than the real data. But I think this is understandable because you will have some assumptions for your model. Next, a very interesting thing is how to use the SEZAN3 to evaluate the goodness of the pseudotime. So here we have the test data, and we also have the pseudotime inferred by the slingshot pseudotime inference algorithm. And what we did here is after we get the pseudotime estimated by slingshot, we add some random perturbation of the inferred pseudotime. So if the perturbation is 100%, then it means that we just randomly shuffling pseudotime. And then you can think the quality of the pseudotime will be very bad. So based on this idea, we use the different pseudotimes with different perturbation levels to generate the simulation data. And we can see, as we can expect, if your pseudotime is very bad, then your simulation is also very bad. But if you can have a good pseudotime, then your simulation will be very realistic compared to the test data. So therefore, we can use SEZAN3 to do this latent covariate quality assessment. For example, how good your pseudotime is or how good your cell type is. Here we show the agreement between the LISI score and the AIC value for each model. And we can see those two metrics are agreeing with each other quite well, and we almost observe a perfect correlation between them. And we can also see that with the increased level of the perturbation, the AIC is increasing, which means that your model is worse and worse, and the LISI value is also decreasing, which means that your simulated data is not that similar to your real data now. We think this is very interesting because as far as we know, this is the first approach for assessing the inferred pseudotime, which we think this is very important because nowadays people often try to do the trajectory of pseudotime analysis. But the problem is that they don't know if their pseudotime is trustful. And here, use our method, you can give a very brief assessment for the quality of the pseudotime. So here we come to the conclusion of the SCGen3 framework. So given the feature matrix and the user-provided cell queries, SCGen3 can simulate realistic new data, which is what we did for SCGen2, but extended to more complicated data types. And it can also evaluate the quality of cell queries, for example, pseudotypes or inferred cell types by clustering. And it can also help you to choose the best model. So if you have different model specification, you can use SCGen3 to choose the best one, which can explain your model. And it can also perform the DE test if you want, because the marginal distribution are fitted by regression models, and each regression model is naturally a DE test for each gene. To clarify the usage of SCGen3 cannot infer the cell covariance. So the cell queries are provided by users, and the SCGen3 model will treat them as the truth. So the R package is still under active development. And if you are interested in this work, please just email me and I can share you a development version. We will release it on GitHub once we submit our manuscript. And here I want to acknowledge my collaborator, Qian Wang, which is a first-year PhD in statistics, and also my advisor, Dr. Jingyi Jessica Li, and also all the other members for the Jessica's lab. Thanks, that's all my results. And I also have supplementary results for the model selection part, but if you are interested in it, I can explain. Thanks.